Hello, this is Dr. James Kupik, Chief Medical Officer at ProMIS Neurosciences. Thank you for clicking on this presentation. I've just returned from the Alzheimer's Association International Conference in Los Angeles, and it was certainly very exciting to see and discuss all the new data on fluid-based biomarkers for neurodegenerative disorders and learn how they will certainly impact clinical development strategy. Here's the agenda for today's presentation. First, I'll discuss the impact of these biomarkers on the early clinical development of potential disease-modifying therapies. And the good news, by the way, is that a number of these brain-based biomarkers can now be assayed in the blood, and this will contribute to ease of sampling and clinical trial speed. Second, I'll give 3 examples to show how selecting an appropriate and focused patient population is critical to maximizing the odds of success in revealing an early assessment of a therapy's true potential. But first, a few basic comments about biomarkers. They are objective, quantitative measurements that reflect some aspect of human physiology or disease, or even confirm how a disease can respond to a particular therapy. The table on the left shows a number of biomarkers that we are all quite familiar with, be it either blood pressure measurements, blood glucose, or cholesterol levels. They have been in use for decades, and they therefore have widespread acceptance by physicians, patients, and regulatory authorities. Biomarkers are used throughout drug development. In clinical studies, we use biomarkers to demonstrate the safety of an investigational compound, to help select ideal patients for a particular study, and we use them to demonstrate that our treatment is having a measurable effect on the pathologic disease process. We call the latter pharmacodynamic biomarkers because they dynamically respond to the pharmacologic action of an investigational drug. The problem in neurologic disorders is that most of the really important biomarkers are large proteins. In years past, we've been severely hampered by the fact it's difficult to accurately measure brain proteins that are excreted into the bloodstream. Blood levels are very low, and the proteins tend to get degraded very rapidly in the blood. Well, that has been the case until just the last few years. New immunologic or mass spectroscopy techniques now permit measurements at 1/100th or 1/1000th the concentration we typically have been able to measure. Let me give you 2 examples of a great new pharmacodynamic biomarker called neurofilament light, or NFL. This protein stabilizes the shape and function of axons. That's the part of the neuron that travels far through the brain tissue to connect and communicate with other neurons. When axons and neurons are damaged or destroyed, NFL goes up in both the cerebral spinal fluid and also in the blood. The correlation of NFL levels between CSF and blood is incredibly and surprisingly good, but NFL is not specific for any particular neurodegenerative disease because it tends to increase in almost all of them, some higher than others. On the left panel, you see that at 6 months after initiation of Tysabri, in a group of patients with multiple sclerosis, the NFL levels went down to normal levels. This occurred at the same time the patients improved clinically. The same is true on the right panel for the young patient administered Spinraza for spinal muscular atrophy. The NFL levels tracked the clinical response and plummeted from a very high level after just a couple of doses. Although NFL in the examples above were measured in CSF, we now know from numerous studies that there is equivalency between CSF and blood-based measurements, which use the new ultra-sensitive assay techniques. Although NFL appears to be ahead of the pack, so to speak, in demonstrating its value to monitor disease and drug response, there are other biomarkers that show great promise. In red, I've circled a few proteins I'm quite interested in. They all reflect a different process inherent to a particular disease. For example, neurogranin is a protein that contributes to dendritic spine structure and function, and is critical for memory consolidation. It's quite elevated in the CSF of patients with Alzheimer's disease, but, surprisingly, not other neurodegenerative disorders. There is a new blood assay for phosphorylated tau that was discussed during a number of scientific sessions at this year's AAIC. Its strength as a biomarker, reflecting neurofibrillary tangle formation or drug response, is just beginning to be understood. SNAP25 reflects activity at the terminus of axons and is clearly modified when diseases attack the synaptic connections of the brain. Yes, there are other cells in the brain, and they are activated in response to disease, such as the microglia and astrocytes, and we can also measure their activity and damage. Biomarkers, both blood and CSF, now allow us to peer inside the brain to assess the damage. In fact, many colleagues in the research community are calling this panoply of various blood and CSF biomarkers a, quote, liquid biopsy, unquote. Well, let's go back to NFL. It's elevated in patients with Alzheimer's disease and correlates with atrophy of brain tissue. NFL levels continue to increase as cognition worsens in Alzheimer's disease. It is elevated in multiple system atrophy, which is an atypical Parkinson-like disorder, and we find very high levels in amyotrophic lateral sclerosis, frontal temporal dementia, various types of stroke, anoxic encephalopathy, and active HIV dementia. A very important paper was published 3 months ago evaluating NFL in patients with Alzheimer's disease, or mild cognitive impairment due to Alzheimer's disease, and they used thousands of samples from the ADNI study. They clearly showed the value of NFL to reflect the underlying degenerative process, and the authors stated that this biomarker may in fact be useful to monitor the effects of disease-modifying drugs in patients with Alzheimer's disease. Well, all of my development friends at other companies that I spoke to during AAIC indicated they were already collecting NFL in their earliest studies. In fact, one key member of the research community called NFL the future, quote, cholesterol of AD, unquote. That may or may not be the case, but at the very least, it's an incredible biomarker with huge potential. If we hypothesize that an effective drug should diminish neuronal death and axonal injury in advance of more typically recognized clinical benefits, well, then we should be able to show early on a reduction in elevated NFL levels. All right, let's shift gears. Let me give you 3 examples where being smart and testing an investigational drug in a disease-specific population should maximize the odds of demonstrating proof of concept early in the development process. Neurodegenerative disorders are caused by misfolded aggregated proteins, and ProMIS has generated highly selective antibody therapies to target these proteins. In each case, abnormal fluid-based biomarkers should demonstrate a correction in response to therapy. Example number 1. MSA is an atypical Parkinson-like disorder that is rare and aggressive. It is caused by misfolded alpha-synuclein, a protein, and it's this same misfolded protein that produces Parkinson's disease and Lewy body dementia. NFL is quite elevated in MSA. Since we now have a blood-based assay, one can measure NFL as frequently as once a month in order to determine how quickly a patient responds to therapy. With a therapy that is truly disease-modifying, one should observe a normalization of NFL over time. ProMIS currently has multiple antibodies demonstrating great selectivity for the toxic misfolded form of alpha-synuclein, and a study in MSA patients could facilitate proof of concept for our antibody as early as phase 1. If the lead compound does not impact NFL or the other biomarkers in such a study, then the feedback loop would be shortened and enable us to test a different antibody targeting a different region on the misfolded protein. Example number 2. Patients with Down syndrome have the highest genetically based incidence of Alzheimer's disease. They have a 3rd copy of chromosome 21, and that chromosome genetically codes for the amyloid precursor protein. Thus, these patients begin to accumulate beta amyloid in their brains earlier and at a faster rate than patients with sporadic Alzheimer's disease. And the large majority of them go on to develop dementia. There are already large cohorts of patients who have been assessed with Down syndrome, as well as a clinical trial infrastructure in the US and in the EU. We plan to work with the Down syndrome research community to assess our lead compound, PMN 310, and determine whether it has the potential to benefit patients. Example number 3. Patients with ALS have significant damage to their axons, and thus very elevated levels of NFL. This is a horrible, aggressive disease, and the majority of patients have pathology consistent with misfolding and aggregation of a protein called TDP43. And by the way, this same protein is misfolded and deposited in patients having frontotemporal dementia, or FTD. ProMIS has a number of highly selective antibodies targeting TDP-43. We know that it strongly binds brain tissue from patients who have died of ALS. So again, by assessing the impact of our antibody therapies on NFL levels in phase 1, we can come to an early decision as to whether to continue to invest in this therapeutic approach or try something different. Okay, I've given you the 3 examples where we can test early in an optimal population. In phase 1, as you can see on this slide, we demonstrate the safety of our therapy at low doses and only escalate up to the next higher dose in a staggered fashion when we're satisfied that the current dose is safe and well tolerated. We typically continue to escalate until we reach the maximum tolerated dose. We will conduct part of our phase 1 studies in a double-blind fashion, then shift to an open-label extension where each individual knows that they are receiving active drug and not a placebo. By designing the phase 1 study this way, we can also collect blood samples for up to 1 year in order to assess biomarkers. There is often existing data for these biomarkers from other sources, sometimes what we call a large natural history database. When one has access to such data, that allows one to compare the change in the biomarker in an interventional study over 12 months and compare that data to what would be expected in that patient population had they not gone on an investigational therapy. A change in the biomarker, particularly a dose-responsive change, would strongly suggest a significant impact on neuronal preservation. Drug development for neurodegenerative disorders is going to look a lot different very soon. In the old model, as you can see on the left, we would frequently move into phase 3 without much phase 2 evidence. A very high-risk decision and a very expensive one, too. The new model of development, as you can see on the right, focuses on early biomarker assessments that will permit a clear go, no-go decision for subsequent development and investment. All this for about $5 to $10 million in phase 1. No longer will we need to wait until a futility analysis is conducted in phase 3 to make such a decision. That model is clearly no longer sustainable. So here's my last slide. I've reviewed the shift in development based upon the new availability of fluid biomarkers and their impact on decision making. I hope I'll have the chance in the future to discuss in more detail some of the other exciting biomarkers that I've listed on this slide. In developing investigational drugs in this field, it is also imperative to select the optimal disease population in order to rapidly test the therapeutic hypothesis. I've given 3 examples that we at ProMIS are considering as we move our portfolio forward. It's really not hyperbole to state that we're in the midst of a transformational drug development revolution that wasn't even imagined 4 to 5 years ago. These new biomarkers have the tremendous potential to measure drug treatment effect. They will facilitate a faster assessment of potential therapies at much lower cost, and this is something that I'm really super excited about. So thank you again for joining me today. This is James Kupik, Chief Medical Officer at ProMIS Neurosciences. I sincerely appreciate your time and attention.